Okay, we start at 18:10. Very good evening, everyone. So we will soon will start our class as usual. Glass there. Ole, ole. Up hade. Glass ta there. Daman, 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 think we can start now. So from the day 4th or 3rd week, I think we are having this. I don't know when we started these classes. So let's continue with this DNA fingerprinting. So why we use DNA fingerprinting? It's mainly used in order to tell individual that belongs to similar species or it's from the different species. So DNA sequence could be very variable and can therefore be used identifying characteristics. So it has various advantages over other sources of evidence like fingerprints, blood type, etc. And they are quite highly accurate and can be gathered from trace scene evidence. So how do we use it? We used, like yesterday we did Rflp, so restriction fragment length polymorphism. So restriction enzymes are molecules that can cut DNA into pieces, and each enzyme cuts at very specific DNA sequence. So while all human beings share roughly the same DNA, so it's quite, but in fact, small number of differences are there, and these differences we can see it with the help of restriction enzyme. So one animation for Rflp, how does the DNA fingerprinting principle works? So here is your BAM H1 restriction enzyme, and this is your sequence GGATCC, and the restriction enzymes comes in and it cut and make your sticky ends. So let's say individual one, and it could cut the DNA from 4 different parts, leading to 4 different groups, fragments of your DNA, or 5, sorry. But in individual 2, 3 restriction enzymes could only cut, so it will give 4 fragments. So when we do the gel electrophoresis, then we can see these 5 versus 4 difference, and we can differentiate between these 2 individuals. So essentially, once the DNA has been cutted out, enzymes that we want to see, various sizes, we can see it. So each individual's binding patterns should be different because restriction enzymes will cut each person's DNA at different points. And we see different fragment sizes which travels through the distances over the gel and after the passing the current through them. So like this, we see the binding of agarose gel electrophoresis, different binding patterns. Yeah, this is the molecular markers at the end, and this is the for different individuals, all having the same different binding patterns. So this is showing 2 double here, 2 separated, 2 separated at different levels. So this DNA fingerprinting is very helpful in the forensic analysis. So induction to DNA fingerprinting and forensics, which can be defined as intersection of law and science. So first photography was done in 1985, yeah, for the DNA fingerprinting. So DNA fingerprinting shows unique patterns from one person to the next and used in paternity disputes and as a forensic evidence. So preparing a DNA fingerprinting. So specimens collections could be licked envelope or dirty laundry, a cigarette butt, and saliva. So special precautions in handling specimens, so gloves, disposable instruments, avoid talking, sneezing, avoid touching samples with your skins, air dry the evidence before packaging, so mold does not grow in them. So enemies of evidence are sunlight, high temperatures, bacteria, moisture. An ideal sample is 1 ml of fresh whole blood treated with EDTA. So Rflp, restriction fragment length polymorphism. Here we can see the nucleotide sequences, variations in regions of DNA that generates fragments length differences according to the presence or absence of restriction enzymes. So here the individual one, it is cutting from two different sites, production of three, here only two, so leading to only two parts here. One is not cutting, so only one is cutting here. Then comes the Rflp fragments that can be separated by gel electrophoresis. So in the individual one, we were able to see three fragments, and individual two, it were two fragments. So that's why we see these things. Is this working? No, it's not working. Then southern blot. This is where we used to hybridize a DNA probe, like we have discussed before. So in the southern blot, first step that is your DNA. We extract the genomic DNA and then run on the agarose gel. Then we transfer this to the nylon membrane. After transferring to the nylon membrane, to hybridize a radioactive labeled DNA probe sequence on the membrane. So membrane, the DNA are transferred. Then the labeled probe that you want to see, let's say from this to this. So this is incubated with the membrane, and once it is there, your radioactively labeled membrane to large sheet of film. You will visualize bands where the hybridized to the DNA. So then comes the variable number tandem repeats, VNTRs, which are the sequence repeated multiple times, and it varies from person to person. So in individual one, it might be repeated 5 times. In individual two, it might be repeated 2 times. GATC, 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 like this. And then they're mostly occurring in introns, and we can amplify with the PCR and then run on the agarose gels to produce unique fragments. So further to amplify it, we use this PCR techniques that we have discussed zillion of times, which is to amplify to segments of DNA. So I want to show you actually, as I used to have a lot of virtual labs actually, but due to DNA extraction, PCR, gel electrophoresis, and many more, we used to have very fun loving classes. But as you can see, our PCR virtual lab interactive was built using Adobe Flash, which is no longer supported in the modern web browsers. We are actively converting our old Flash pieces into new formats that will support it long in the future. We hope to have virtual lab back in spring 2021. When is the spring 2021? Is it now? I think so. When it is? Because I really want these virtual workshops for all of you. Oh, it's already started from 20th March and it will end till 21st of June. Okay, good. Good to know, good to know. Because here if you click on the virtual labs, but I will share this link with all of you. So whenever they are available, you can access them from this link. At the moment, we are shut down for unknown reason. For the known reason is just Adobe Photoshop. Neither flow cytometry is working, neither DNA microarray is working, nothing is working. This was DNA extraction was such a nice lab actually. This was exactly how we do it in the lab. And this is also not working. So sorry for that. So PCR we have discussed zillion times. We have DNA to be amplified, primers, DNA polymerase, nucleotides, mix them together, have your thermocycler, have different temperatures of denaturing of 94, annealing for 50 to 60, then elongation at 72, then do it for 35 to 30 times, heat, mine, copy. and then you have these cycles 1, 2, 3, 4, 5, and then you're gonna have amplification. Then you can also use this technique of DNA fingerprinting for diagnostic diseases. For, let's say, this is normal, this is disease state, recessive one, and this is the carrier state. So when you cut them with restriction enzymes, in the normal case you see the band pair of only 376, but in the case of disease you see 201 and 175 base pairs, and the carrier also have 376 and these both. So in this way you can differentiate between them. Also, you can check between your parents, mother and father, how the siblings are, how things are normal and diseased ones. Veterinary testings are one of the applications for forensic science, and then genealogy. There is one animation, maybe this is helpful. So on the evening of November 1st, at approximately 8:15 p.m., Jimmy Sweet entered his bedroom and walked over at his desk and sat down at his computer. So while reaching for the computer switch, he noticed out of the corner of his eye that one of the items on typically well organized shelf was out of place. Jimmy shot across the room for closer examination. Sure enough, the object in the question had indeed been disturbed. The object had been sealed in airtight package, and the package was now ripped open. The object was still inside, but it was no longer its original conditions. So in Jimmy's eyes, it was now worthless. So Jimmy pulled out, and what happened? His most valued possession, his holographic Nova lollipop. The confectionery treat was now a sticky mess. Someone had obviously indulged him or herself with sugary molecules. So lollipop holographic image had been licked away. Oh, what a serious crime is there. Okay, now who was the culprit? Suspect. The primary suspect in this case are Jimmy's 7 sisters. Oh man. Candy, cookie, sugar, lolly, honey, brandy, and caramela. Each one of these sisters is notorious and candy lover, as the name suggests, and easily capable of committing these crimes of confection. Okay, and it's a funny one. These suspects have been detained, and DNA fingerprints of each are available. So what do we need in this? So fortunately for this case, a lollipop cannot be licked without leaving behind a bit of saliva. Oh, so some of the saliva will be there on the candy. So our task as a Nova Lab chief technician is to create a DNA fingerprint from the saliva left on the lollipop, and we should use it to find out the culprit of this crime. So this is a virtual lab. We are lucky. We have a virtual lab finally. So let's add precision enzymes to the DNA samples. How we do it? So this is our restriction enzyme. So they have been added to the DNA. So we have just added restriction enzymes to DNA samples, and they work like a scissors cutting the long DNA molecules at different locations. Where those scissors cut depends on the code within the DNA molecules and the code within the enzymes. For example, so one type of enzyme will encounter a sequence of GATTC. The length of these fragments will vary from person to person. So some fragments will be longer, for some shorter. So it will cut according to the different persons. Now we pour agarose gel into tray on lab counter. So this is our agarose gel. We put this over the, I think, on this. Yeah. So we just have poured this agarose gel over the tray, which is a thick porous jello-like substance, and it will act like as a molecular stainer and allowing smaller pieces of DNA to move through more easily than the larger pieces. So now we will pour this DNA over, not like this, we pour DNA. This is not the way, scientifically. We power on it, then our DNA will move. So this is not the across-gel electrophoresis run. Believe me, this is not the way. I will teach you in upcoming sessions how we run it. So our DNA with the higher fragments, they will be on up, and with the smaller fragments, they will be at the bottom. And then we put nylon membrane on top so that they could be come over this nylon membrane or your gel patterns. So we'll just transfer them. What's now? One more. And then we add maybe probes to the nylon membrane. So after that, these probes of the nylon membrane of the DNA that have been radioactively labeled, and they will attach them to the DNA fragments, and they will attach mainly to those codes that we are looking for, and the excess are washed away, and we put this over the X-ray film. So we have just, and now we will expose the X-ray, and we will see into the developer. Now X-ray has been developed. The film displays the locations of nylon membrane where the probe attached, and this is our DNA fragments. So choose the culprit. So here is the saliva of the DNA. On the right hand side looks like this is the saliva, and these are 7 sisters whose DNA fragments looks like this. So we need to find the suspect. So if we see it from the, so what pattern we see? We see 3 patterns here. Where do we see these 3 patterns like this, and then big pattern? 3 small patterns first, and then big pattern, then big pattern, and then small patterns. Yes, she is a culprit. Honey was the one who did that. So honey was a culprit. So I will put this on this. So see, it's matching. So that's how we match them, and then we find she's the one, honey. So we have correctly determined the honey is a culprit, and you're most welcome to do the advanced cases in the future, but this case is being solved now. So thank you very much. So this was very lovely workshop actually. I did this for the first time also. I was not aware of it. Very nice. So this is how victim, crime scene, suspect. So like this we identify them. So chronology, some applications. So that's it. We are done from the detailed DNA fingerprinting for today. So yeah. So we are done for the DNA-based markers, friction enzymes, and proper way of doing DNA fingerprinting. I hope this is all clear for today. Now we come to the next big part, that is protein. Protein isolations, purification, that's more over your biochemistry and proteomics part actually. This is I'm always excited to teach you all these cases. So let's do it. Thank you, Vivek Shukla. So let's have a 5 minutes, 1 minute break or 2 minutes break. Then we start with a fresh start for this. Let's continue. So, protein isolation, quantification, and purification. This is a quite big one, big topic here. Maybe we finish in 3 days or 2 days. Let's see how it works. But I think our course need to be finished by the next week. So today is 27th. So I will try to finish it by 7th or 9th. So let's see how far we could go. So we are today on around 50% of your task. So protein isolations, quantification, purification. This part has been divided into homogenization, fractionation, centrifugation, quantitation, chromatography, and electrophoresis. So Nobel Prize winners on the chromatography and electrography and chromatography and electrophoresis. So some of the Nobel Prize winners here: Archer John Porter, Richard Lawrence Millingstons, Arne William Corinne Tisles. So they are all known for the invention of the partition chromatography. This brought a big thing into the market. And then this Tislius from the Sweden Uppsala University, Sweden, he identified electrophoresis, adsorption analysis, and complex nature of serum proteins. So they discovered many big things in their life. Then comes Nobel Prize winners in the field of mass spectroscopy and sedimentation. So John B.F. in 2002, he identified methods for identifications of structure analysis of biological molecules. Then Koichi Tanaka from Japan, he found out the soft desorption ionization methods for mass spectrometry analysis of biological macromolecules. Then Theodor Swedberg for his dispersed systems. His Swedberg's equation is there famous from his name. Then organelle fractionations, that is to differentiate between your different organelle. So it was shared by 3 scientists concerning the structural and functional organization of the cells: Albert Claude, Christian de Douai, George E. Palladay. So one from School of Medicine, New Haven, Yale University, Rockefeller University, and Université Catholique de Leuven, Belgium. Then comes your famous Frederick Sanger from 1958 from the University of Cambridge for the work on the protein sequencing. So these were the famous people who played a quite important role in the field of protein biochemistry. And now let's start with the first part of this series of today, that is your protein isolation. So homogenization, which is the first step, and it's a disrupting cells. So and in the fractionations, crude separations of homogenates into fractionation is done. So homogenizations include Downs homogenizer, sonication, osmotic shock, and French press. So homogenate forms are the one which centrifuge at 500 G for 10 minutes. And then the supernatant that we got, then the palette, that we got will have nuclear fractions. Then we again go for 10,000 G centrifugation for 20 minutes. Then the palette has your mitochondrial fractionation, whereas your supernatant will have again for 1000 G for 1 hour. So the palette will have microsomal fractions, and the upper part supernatant will be cytosol soluble proteins in it. So by this way we can differentiate between different patterns with different G's, and we can have different cellular organelles. Then comes how we can isolate these proteins. So there's two methods, by differential centrifugation and rate zonal centrifugation. So in the differential centrifugations, we have more dense particles and less dense particles. Yeah, we have mixture of both particles, and then we centrifuge them, the palette, the more dense will be down and the less dense will be in the supernatant, and we can differentiate between them. Otherwise, we can have a sucrose gradient, and then we have smaller particle and larger particles mixed together. And then what will happen after some centrifugation, that you will see two layers of the dense particle and lighter dense particle, and with the help of pipette you can separate them. So these are the various centrifugation speeds in the palettes that we can separate our cellular organelles. So for 3000 G for 5 minutes, large organelles could be getted out, and at 10,000 G for 10 minutes, mitochondrial inclusion bodies. Then at 40,000 G for 30 minutes, membrane, plasma membrane, tonoplast could be getted out. And at 200,000 G for 16 hours, we can have the large protein complexes. Whereas the 400,000 G for 24 hours, so we can have proteins of 50,000 Dalton. So sedimentation velocity technique, here what we can see is that there is a low density solution on the left and high density solution on the right. And then after the centrifugation, we can centrifuge these tubes, density gradients, and then we layer these samples, and then this is a separation due to the separations of sedimentation coefficients. We have a fractionations collected like this. Then how to quantify your protein? Once you have isolated your proteins, you have your proteins with you after the fractionation and centrifugation. Just a second. It's time to quantify that how much protein have you got at particular time. I mean, for the DNA isolated, we can do it simply by spectroscopy, the spectrophotometer. You add your DNA, it will tell you this in nanogram, microgram, how much DNA you have produced. But for the case of protein, it cannot be do the same way. Though yes, it's a spectrophotometer, but it's indirect method by the Lowry. Bradford, initially we used to have Lowry method. But at the moment, Bradford protein determination method is the best one, which we do with the Comesi Blue G250 dye, which is readily to organelle and lysyl residues of proteins. So this is the Bradford assay. How we do it?
In this video, you will learn how to perform protein quantitation using the Bradford assay. Protein quantitation is one of the most commonly performed procedures in a biotechnology laboratory. In this activity, the protein concentration in milk will be measured using the Bradford assay. For this activity, you will need 1x Bradford reagent and 1x phosphate buffered saline, or PBS. Using a 20 to 200 microliter micropipette, set the pipette to 98 microliters. Pipette 98 microliters of 1x PBS into an empty microcentrifuge tube labeled sample. Switch to a 2 to 20 microliter micropipette and set the pipette to 2 microliters. Pipette 2 microliters of milk sample into the microtube labeled Sample A. Mix the milk sample and PBS by either pipetting or vortexing. This mixture has diluted the milk to 1 part in 50. Label 2 cuvettes, one control and the other sample. Do not handle the cuvette where the light passes through, and make certain your labels are well above this area. Pipette 20 microliters of diluted milk sample into the cuvette labeled sample, and 20 microliters of 1x PBS into the cuvette labeled control. Next, we will set up protein standards. See your protocol for the correct concentrations. Label a cuvette for each standard and mark it with the corresponding concentration. Next, add 20 microliters of each protein standard into the corresponding cuvette. Make certain you use a clean pipette tip for each sample. Now you are ready to add the Bradford reagent to all of the cuvettes you have prepared, including the sample and control. Add 1 milliliter of 1x Bradford reagent to each of the cuvettes. Mix completely by pipetting up and down with the micropipette. Incubate the cuvettes at room temperature for 5 minutes. After 5 minutes, visually compare the cuvette containing the milk sample to the cuvettes containing the protein standards. Determine the standard that most closely matches the color of the milk sample. Estimate the protein concentration of the milk sample based upon the visual comparison. To determine the protein concentration using a spectrophotometer, select for the Bradford assay. When asked to insert blank into the spectrophotometer, insert the cuvette labeled control. Use the blank to set the spectrophotometer to 0 absorbance, or 100% transmittance. If instructed, read the absorbance of the 7 protein standards and record the absorbance values. Remove the cuvette labeled blank and insert the milk sample into the spectrophotometer. Read the absorbance and record the value.
So that was the how we do the Bradford assay, and then is a protein quantitation method in which we add a sample dye to it, heat for 10 minutes, let it cool temperature, and then we read the fluorescence from your samples. This is also another method which was shown in the bar techniques in 2003. Then comes the ultra sensitive fluorescence based protein quantitation. Here we can see that in this method you have fluorescence on the y-axis and protein on the x-axis is microgram per ml, and then we see this linear graph. Then whatever the fluorescence that we read will be the protein concentration in microgram per ml. Now comes the next method. Now we have done the isolation, we have done the quantitations. Now it comes to purify your protein, because it will have many things that unwanted that we do not want to be part of our protein sample. It has to be pure protein. So in order to do so, some techniques we will discuss in protein purifications. So a technique used to remove small molecules from the protein solution is to change the buffer compositions of protein solutions. And dialysis tubing or bag is made of cellulose and has different cutoff range. So here we have a dialysis bag. We have closed it from the top and bottom. We have a buffer solution, concentrated solution, and dialysis bag inside. So in that we have mixture of big and small molecules. And then at equilibrium, what we see is yeah, that your small particles are removed already and you have large particles inside only. So let's see how we do this dialysis of protein. So next methods for the protein purifications are chromatography, which includes mainly chromatographic methods. A mobile phase is liquid, and stationary phase could be liquid or solid in this. So let's talk about first chromatography technique, that is your gel filtration chromatography. So in gel filtration chromatography, so you have a protein sample here and you have a gel at the bottom. Actually in the hole and this silica gel, molecular exclusion gel. So with the time, what you will see that there are some carbohydrate polymers beads where your small molecules will get enter and attach to them, and the bigger ones will start to flow. So like the yellow one will flow fast, green one will do it later than the red ones. So separations of proteins, here we have large and small molecules, and they are being separated via polymer bed beads. So let's check this technique, how we do this.
We're starting with gel filtration medium. This is Cefidex G50. The number here gives you an approximation of the size exclusion. So this is will exclude proteins above 50 kilodaltons, 50,000 molecular weight. Inside is a white powder. In the beaker I put 9 grams of the G50 powder, and I swelled the powder overnight using this tris buffer, 30 millimolar tris buffer, pH 8.0. You can see that the 9 grams has swollen up to a volume of about 90 milliliters, almost 10 milliliters per gram of solid. Now we're going to gently stir the gel. It's important not to stir this too vigorously. Don't mechanically stir this. That can break your gel particles up into very fine dust, which will impede your flow rate in the column. This is making a slurry. I'm using a sawed-off pipette to transfer my slurry to the column. For gel filtration, I want a fairly tall gel bed. So I'm trying to mostly fill this column. And excess buffer will drip out the bottom. So here you can see the drips coming out the bottom. Gel bed is starting to settle a little bit. Gel bed is right here. I'll add a little more buffer so that it doesn't run dry at the top. So I'm gently adding a little buffer to the top. This looks great. You can see the gel bed is fully settled here. There's a little bit of buffer on the top. I've capped the bottom right now to keep it from going dry. I'm going to put a mixture of 2 molecules onto the column. Blue dextran is a very large molecule. It's a polysaccharide. Molecular weight about 1 million. Fluorescine, on the other hand, is a small molecule. Molecular weight about 350 grams per mole. I chose these 2 molecules because they're nicely colored, so I'll be easily able to see the separation visually as they go through the gel filtration. I've drained the buffer, so now there's no residual buffer on top of the gel, and I've made a mixture of a total of 0.8 milliliters, 400 microliters of the fluorescine, 400 microliters of the blue dextran. Nice green color. I'll separate these on my gel. For this size column, this is getting near the maximum I would put on the column. I'm using a Pasteur pipette to gently load my material onto the column without disturbing the gel bed. So here you can see the blue molecules, the large molecules, are moving faster through the gel. So I'll hope that I can get a complete separation between the blue and the yellow. Now the excess on the top of the gel is all soaked in. So I'm adding buffer very carefully to the top of my gel filtration column. I'll just add a fraction of a milliliter first to help get everything into the gel before I add a large volume of buffer. So now the separation is proceeded somewhat. I'm going to try to add a decently large volume this time of buffer on the top. About a milliliter would be great. If I do this well, then the buffer that I put on the top remains colorless because all the colored molecules have already gone into the gel. Alright, now you can start to see a little colorless layer on the top. That's perfect. All the colored molecules are in the G50, and they're starting to separate quite nicely. The yellow molecules are up here, the blue are down here. Still got a little overlap. So I may have overloaded this column, but we'll see. I'm hoping that before the blue molecules elute that they'll actually be separated from the yellow, but it's going to be close. So out of the bottom now, blue liquid is dripping. You can see I do have a colorless layer starting to develop here. The fluorescein has been slowed down by the gel filtration. The blue dextran is coming through. So this technique is often used to separate a protein, which is large, from the small molecules that could be salts or could be a fluorescent tag that is unwanted. I've topped up with buffer on the top so that it never runs dry. The fluorescein is now eluting from the column. Fluorescein is fluorescent. I'm shining a little blue light, UV light on it, and you can see it glows green. The last little bits of fluorescein are emerging from the column now. Here's the fractionation. This first tube has nothing in it. So this volume, which looks to me about 2.5 milliliters, is called a void volume, where nothing comes out. But then the blue molecules, the blue dextran, came through the column swiftly. So this is about a milliliter. Some of the blue dextran tailed into the next fraction. This is low concentration. Then I have a nearly colorless fraction here, and then the fluorescein starts coming out here. Fluorescein stopped coming out right about here. So the total volume will be 2.5, 3.5, 4.5, 5, 6, 7, 8, 9, about 10 milliliters is the total volume, which is what I would expect for a column of this size. Often what you want is just this first fraction with the large molecules. If you're trying to desalt a protein or remove the unbound label from a protein, then this is the fraction that has value. These fractions would just be junk. The original mixture, you can see, glows nicely. And what I believe is isolated blue dextran from the column has no glow in the UV. That's a sign that I have successfully separated out all the fluorescein from the blue dextran. So the column, if I've done it well, comes out completely colorless. It's reusable. Sometimes microorganisms like to grow in the cephadex, so if you want to preserve your column for a long period, keep it in the fridge, certainly. And sometimes azide is added to the buffer to prevent growth of microorganisms. Here's tris buffer with 0.01% sodium azide, and this will prevent microorganisms from chewing on the dextran that's in the column if you're storing it over a long period of time.
So I think that's enough for today. I will not take the another part of our purification method. So tomorrow we will do with the ion exchange chromatography. For today, that's it, because time will not allow us more. But sonication is like with the sound we disrupt your cells. The cell wall are being disrupted with the sonication. So we have our cells here in the Eppendorf tube. We have to add the cell lysis buffer, which will break your cells. And this is the sonicator. And we put this sample tube and press the start button. It will create a lot of sound and voice clean it with ethanol, and that's it. So mainly with the help of sound, we can high frequency wave, we can finish this up. And anyone from Delhi, um, if anyone out of you is from Delhi, please let me know if you can get the oxygen from where we can get it. I will really appreciate that. Some of my friends need it. Her mother needs it. So please let me know. Thank you very much. That's it for today.